So this is going to be quite an interesting lecture from to me. It may seem a little out there to you all, but it turns out to be pretty useful. So what this is going to be about is topicity and determining chirotopicity or chirotopic points. Okay, so what this is let's just break this down. So topicity refers to a description of atoms, groups, or side chains in a molecule, basically with a region, with something as big as an entire half of a molecule, as big as a giant ring portion of a molecule, just as long as it's not the whole molecule, or as small as a single atom, we can define what's called the topicity. Now, what this is really useful for is when looking in NMR, two H atoms or multiple H atoms will have the same NMR properties if they're in the same chemical environment. Okay, let's look at an example of this. All right, so this is just a brief review of NMR. Let's say we've got these two hydrogens. Okay, if we wanted to count the signals for this, we would look at all the hydrogens that are in different environments, and we'd say, well, the three that are on the CH3 at the end are in the same chemical environment. The next two, which are the ones I've got written explicitly, those are in the same chemical environment. Let's call them B and B if this is HA. And we could keep going, and we could look at the aromatic ring. We could count all the signals. The important thing is that in this example, both of the hydrogens that I've labeled HB are in the same chemical environment. They're both attached to a carbon that's attached to a CH3 that's attached to a CH2 on the opposite side. Okay, what happens though if I introduce a chiral center, a stereocenter? Let's change this to let's put in a amine group here or something. Okay. Now, it's not hydrogen, which I'll label C. It's not hydrogen C I'm worried about. Hydrogen C is part of the stereocenter. Rather, I'm worried about the hydrogens that aren't part of the stereocenter. That is, I'm going to go back to these two hydrogens. So it's all about being in the same chemical environment. And I loosely describe that as, okay, the hydrogens in this first molecule I drew, one's attached to a CH3, the other's attached to a CH2. More broadly, we could say that the HBs, both HBs are equidistant from the HCs. So the HB that's drawn with a wedge, that's equidistant to HC, or that's the same distance away from HC with a wedge as HB with a dash is from HC with a dash. That is, if I wanted to compare and say, well, HB is really far away from HC down here as a dash compared to where this HB is, you would be like, well, that doesn't matter because, well, HB is close to this HC. The dashed HB is equidistant to, I mean, it's the same distance, you know, it's got the same, it's in the same chemical environment. No matter how you spin it, it's hard to spin or quantify the chemical environment. Okay, so what we do though, or what happens when we have a stereocenter is now we say, ooh, now it's complicated. even though we're not part of the ring, even though the CH2 should be the same, this green CH2 is closer to the NH2 than the yellow H is. Okay, now what if I rotate it? Well, if I rotate it, I'm in a similar situation where H is closer to something that's different than the other thing. By introducing the stereocenter, you take groups that are otherwise not affected by 4 different things attached to carbon, okay? And you make them now in different chemical environments, which means we're going to be different numbers of signals and the NMR. All of a sudden, we have like HB and HB, call it prime. Okay, so HB and HB prime are in different chemical environments, resulting in different NMR signals. They're no longer the same. And this is all due to the chiral center, due to the stereocenter. Now, what if we have a racemic mixture at that NH2? Well, just to explain briefly, racemic, 2 enantiomers combine 1-to-1 to make a racemic mixture. So if I don't know that I've got the NH2 as a dash, if I've got both of them present, the wedge and the dash, you observe the same effect. You observe that HB and HB prime are in different chemical environments. What if the NH2 is a dash? Isn't HB prime then the same as HB in the original molecule? Yes, enantiomers have the same properties. So if we look at one by itself, it's also going to emerge with these properties. Point is, racemic mixtures don't matter. So it's true of racemates or racemic mixtures and homochiral samples. Okay, so just ignore the idea of a racemic mixture and a homochiral sample. You'll see this no matter what. It's pretty telling when you get to the NMR data. Now, what about the CH3? Okay, CH3 is kind of interesting. Let's consider an alternative molecule where I just lop out What if I consider an alternative molecule where now we've got a CH3 present in the molecule? What happens there? Well, it turns out that with the CH3, all of the H's are in the same chemical environment, because you might say, well, in this original depiction, that one with the wedge is close to the one with the closer to the NH2 than the one with the dash. What you can do though is rotate so that now the HA that was a wedge is now the HA that's pointing down with a straight line. Due to free rotation, you can actually average the effect of being closer in for some conformations to the NH2. And as a result, for this particular group, these are all in the same chiral environment. It's not affected. Now this is NMR. This is how we determine the structure of our molecules with great accuracy. Okay, right? How many NMRs did we run in organic 2? It was like every time, right? So all the best labs anyway. I don't mean that. Everybody loves the computational lab, right? All the same chemical environment here. So we, even if it only affects NMR, and it doesn't, but even if it only affects NMR, it would be really good to know how we can predict this. Okay, because doing the free rotation in our mind gets to be troublesome. Well, what you can do is rely on the chirotopicity. Determining the chirotopicity will tell you if 2 H's or 2 anythings, groups, are in the same chiral environment or in the same chemical environment. Chiral kind of works, so I'll keep it loosely crossed out. Okay, so determining what you do is you replace or locate the groups, the atoms, groups, whatever. It could be as big as a half of a molecule, just as long as you've got at least 2 of them. And it's usually just 2 of them. So locate the atoms or groups of interest. So in our example, we had okay. What we can do is we can locate the two hydrogens. Boom, there's our two hydrogens. What I want to do is replace one with a test group. Now, this is totally a thought experiment, but it will give you the answer as to whether or not you expect them to be in the same chemical environment. So replace them with a test group. This is usually an isotope. So like 2H, which is deuterium, or 13C. Why am I doing this? Don't worry about it. This is just a thought experiment. It's kind of allowing us to solve the puzzle without actually having to do an experiment. So I'm just going to replace maybe the front one. Well, actually, I'll leave the front one alone. I'll replace the back one with a deuterium, which I'll write as D as opposed to 2H. Okay. Now, this is the new molecule. What I need to do in step 3 here is ask, did I form something with diastereomers? Did I form something with enantiomers? Or did I form something without, I guess, without stereoisomers? Did I form a molecule that has the potential to form diastereomers, enantiomers, or something else? Well, it turns out that we're right here. Enantiomers, or excuse me, diastereomers, because this molecule we could introduce diastereomers, that is, if you flip them. So what I like to do, let's clarify this a second. Let's replace one with the test group and then draw the other one with the test group in the opposite position. This should clear things up. Okay, so we're going to draw two potential molecules. Sorry, I'm going kind of fast through this, but I just realized this is an easier way to learn this. What I'm saying is we draw a circumstance where the dash is a deuterium and the wedge is unreplaced. And then we draw what I've just drawn, a circumstance where the wedge is replaced and the dashed is still a hydrogen. Now what I want to do is compare and ask, did I form, let's get rid of this something with, this just makes it so much easier. I can't believe I missed this all these years. Diastereomers, enantiomers, or a molecule without stereoisomers. Let's call that the same outcome. Oh my gosh, this works so much better. Okay, let's get this back up in front of us. What did we form? Compare these two molecules that I'll highlight in green. I'm so excited about this now. What did we form? Well, if you look, the two molecules have the same composition, connectivity, and everything, but we flipped one of the wedges and dashes. Those two are diastereomers. If they're diastereomers, they're diastereotopic. So that is the topicity, or chiral topicity. It equals diastereotopic. We would label those two hydrogens as being diastereotopic. Enantiomers, this is not the outcome, so I'm going to put no here. Then we have enantiotopic. topic. Without stereocenters, or without stereoisomers, we say homotopic. Without stereoisomers, or the same outcome, we say homotopic. So those are the three possibilities: diastereotopic, enantiotopic, and homotopic. Okay, now identifying that and labeling them will give you insight as to whether or not the two groups that you replace with the test groups are going to be in different chemical environments by NMR. Okay, so let's look at some examples with enantiotopic. We have an example where it's diastereotopic. What about enantiotopic? Well, that'll be this second example. So I'm going to have a break here. I'm going to have some examples. Okay, so we have an NH2 here. What if we replace, we have HA, and I'm going to replace I'm going to keep the wedge the same and replace the dash with a D. Now I'm going to do the same thing. So I go, but I'm going to have the wedge be the deuterium and the dash be the hydrogen. And now if you compare these two together, the issue with both of these structures is that, well, we have a wedge and a dash with the deuterium and the hydrogen. The two hydrogens on the leftmost carbon in either case are the same. So this carbon is not a stereocenter. So flipping it from a wedge to a dash does nothing. So what's the outcome here? Well, the outcome here is we have the same molecule just drawn as a different conformation. And so we would say this is homotopic. Homotopic. Okay, so we have enantiotopic, diastereotopic, and homotopic. We'll do an enantiotopic one at the end. So what does all this mean? Here's the key takeaway. If the groups are diastereotopic, the groups are in different chemical environments. So they're in different chemical environments. And that's going to result in some unusual NMR properties. Let's do an enantiotopic example. Oh, let's finish our thought. Excuse me, sorry. Homotopic and enantiotopic groups are always, I should be careful with the word always. I want to say always. I want you to write always, but I'm going to cross that out and say are in the same chemical environment. Okay, so it's only if it's diastereotopic that you have to worry about unusual NMR properties. Here, we would get the same, how about this, usual or common NMR properties. So you wouldn't see anything unusual. Doublets will look like doublets, triplets like triplets, quartets like quartets, no issues. Whereas if we have a diastereotopic group, things start to get unusual. Maybe we'll look at examples of that in a future lecture. Okay, what I want to do is just talk about an enantiotopic example. Okay, so with an enantiotopic example, what we have is actually our first example where I don't have the NH2 drawn, and I said, what about these two groups here? So what about those two groups? Well, these two groups, if we do the test, we could have one where the wedge is a deuterium, one where the dash is a deuterium, and compare the two. We see that these two now have one stereocenter, and that stereocenter is, and as a result, if we compare these two structures, we flipped the only stereocenter that was present, that is enantiomers. Okay, so what's important to note is that when we introduce that test group, if it's on a CH2, it will introduce a stereocenter, and then we're looking at the impact. If there was already a stereocenter present, then we have a second stereocenter, and we automatically have a diastereotopic group. If, so it's diastereotopic. If we don't have a stereocenter present, we would get to an enantiotopic group. If our test group substitution does not generate a stereocenter, then we have a homotopic group. Okay, it doesn't just have, I'm going to conclude by saying it doesn't just have to be, doesn't just apply to H atoms. Okay, let's look at an amino acid valine. And I'm going to ask, what is the methyl group topicity? So we're looking at the entire methyl group here. So there's a few ways you could do this. Sometimes you say you could take one of the methyl groups and convert it into a CD3 group, or you could take it and convert it into a, you can convert it into a CD3 group. You could convert it into a C13 group. Whatever you want to do, you just have to make it different from the other one and then do the same substitution at the opposite position. So the thing to note, though, is we are at a tetrahedral center. So we have to kind of consider wedges and dashes and that sort of thing. So here's the amino acid valine. I'm going to consider one where we put a C13 labeled carbon at the wedge versus having the C13 labeled group as the straight line. Now, I always talk about when we compare stereocenters and we flip them, that we should flip wedge and dash. But going from wedge to straight line or straight line to dash or whatever, just going from wedge to something else, straight line or dash, will get you to a flipped stereocenter. So when I compare these two, I would see that the stereocenter has flipped. Just one stereocenter has flipped, not both. So these are in fact diastereomers. And if they're diastereomers, the methyls are diastereotopic. Okay, so that'll do it for this lecture. Go ahead and close the screen sharing. For the last lecture of week 1, what I'll do is probably just pull a few examples of NMR data that shows the effect of the influence of diastereotopic groups. Okay, have a great day, everyone.